globally renowned for having the most diverse and species-rich flora. This area is also known to have strophic conditions with sandstone soils, regular winter rainfall, frequent fires, which all lead to severe nitrogen deficiency in the soils. And in fact, some of the lowest nitrogen and phosphate levels have been measured in this region. Some of the lowest values worldwide have been recorded from this region. In order to overcome this problem, many of the most ecologically dominant lineages have formed associations with beneficial microorganisms. And some of these organisms include nodulating bacteria, mycorrhizal fungi, and plant growth promoting bacteria. Geophytes in the Cape are also regarded as an ecologically dominant group, but to date, and to our knowledge, no symbioses between any microorganisms and geophytes have been recorded from the Cape. So geophytes have undergone an extensive radiation within the Cape, and one group in particular, which is the Oberlander that we study, has diversified really well, and they include up to 230 species. As far as we know, their evolutionary success in the Cape has been attributed to their unique life history. As far as we know, this includes their geophytic nature, their winter flowering during the wet seasons of the Cape, and their very variable seed germination strategies, which include dormant, intermediate, and recalcitrant seeds. About 40% of the Cape genus have dormant seeds, and I'm sure you all are familiar with dormant seeds. So they germinate by making a hypocotyl anteroot, which emerge and grows and elongates, and at a later stage, cotyledons unfold and foliar leaves develop, that's the common way that all angiosperms germinate. But in stark contrast to this, 60% of the Oberlander genus from the Cape have these very special recalcitrant seeds. So recalcitrant seeds don't undergo any dormancy. They're shed without a lignified integument, and they have no endosperm. So as you can see from these bottom pictures, the recalcitrant seeds have green, probably photosynthetic cotyledons, and as soon as they're shed, these seedlings start to grow. So the cotyledons unfold, and the foliar leaves develop. All this time, they grow and develop without having well-established roots. So from these bottom images, you can see the proportion of cotyledons and foliar leaf material is relatively large without having any roots. So seeing this in so many species from the Cape, we have been extremely puzzled by this, and we think that the most likely explanation would be that these plants have some sort of symbiosis with beneficial microorganisms. So we set out to determine if we can detect any symbioses. We wanted to assess the microbial community composition and see if we could elucidate the nature of the relationships between the plants and possible endophytes. So to do this, we studied delta 15N nitrogen isotope data, and so far we've been using isolation culture techniques to isolate endophytic microbes from sterilized plant material. We've been looking at both vegetative and reproductive plant material, and we've selected 6 phylogenetically representative Oberlander species, which are 3 are dormant species and 3 are recalcitrant, just to represent a good spread of species from 3 locations within the Cape region. So we've surface sterilized these plants, plated them out onto various culture media, grown colonies, and then we've sequenced the 16S region to try and identify these endophytes. At the moment, we're running a very exciting metabarcoding project to see if we can obtain even more in-depth information. So the nitrogen isotope data did reveal a clear difference between dormant and recalcitrant species. It was a significant difference. But as you can see, both of these groups of plants have relatively wide ranges of nitrogen isotope data. what we were interested in were these lower values, or we refer to them as lighter values, which suggests that there might be nitrogen fixation taking place. And we've also been successful in isolating bacterial endophytes from these plants. And so far we've identified 9 different bacterial species from plant material. These species all belong to the genus Bacillus, which are relatively common bacteria, and they're commonly known endophytic bacteria. Very excitingly, all 9 species have known and documented endophytic properties from plants, and they've been described to have plant growth promoting traits and nitrogen fixing abilities. So far, from our 3 sampling sites, there seems to be some specificity depending on where we've sampled plants, but there's also a relatively large amount of overlap among localities from that top figure. And we've seen the same pattern, general pattern, lacking specificity among both the dormant and the recalcitrant plants and individual host species in particular. In this image, the larger squares represent pooled information representing endophytes isolated from various plant organs, and we've sampled root material, bulbs, stems, leaves, and seeds. So a very exciting result was the fact that we found these beneficial bacteria within surface sterilized seed material. So we now know, based on what we've read in the literature, that the bacteria in the seeds have nitrogen fixing properties and plant growth promoting traits. And we've seen this using both our culture-based methods, and we've done fluorescent microscopy, where we've cryo-sectioned seed material and then used fluorescent dyes to visualize the bacteria within the seeds. Even more exciting aspect to all of this is the fact that 3 of the most common bacterial endophytes that we've isolated from almost every single host plant that we've studied so far are that what we've read in the literature are that these bacteria have known oxalotrophic properties. So oxalotrophic bacteria are regarded as relatively rare, and these bacteria are unique because they have the ability of using oxalates as their sole carbon source. The group Oxalis, as the name suggests, represents the relatively abundant source of oxalates or oxalic acid. So this seems like it's almost too good to be true. Very, very cool coincidence. And from my background, from my previous work, and other students from our research lab, we've been studying the morphology, anatomy, and physiology of the Oxalis plants. And we've frequently observed that there are these very well-defined cavities and crypts within the various plant materials. So from these figures, these are just examples showing cavities that we've seen among bulbs, leaves, fruit capsules, and even seeds. So these cavities, some of them are just, they're called ideoblasts, so they're almost like swollen cells with specialized functions. Some of these cavities have very well-defined epithelial linings. So there are different types of cavities, but they're very common. And these images have been obtained through doing wax embedding of material, but in almost all the cases we have seen very clear examples of oxalic acid, crystals, or oxalates present within these cavities. So at the moment, we're thinking that we've detected this highly specialized symbiosis between these endophytic oxalotrophic bacteria, and we think that they live within these cavities, where they feed on the oxalic acid, and in turn they fix nitrogen for the oxalis host plants. And to conclude, these exciting findings with this possible very intimate association and the fact that these bacteria are vertically transmitted leads us to think that all of these traits could be a key mechanism allowing evolutionary success and diversity of this group of plants within the very nutrient depleted region of the Cape.
We have time for questions.
Maybe I missed it, but was there a real difference between the recalcitrant and the normal seeds and the presence of these endophytes?
So we were expecting a very clear difference, but surprisingly not. So we think that these oxalis plants are almost like universal acceptors, where they can just take up whatever is available in the environment. So those ubiquitous endophytes I was talking about, they were present in all species. So,
Great talk. It's very interesting.
thank you.
Oxalis pescum, right? Is ubiquitous in California,
Yes.
and I think it's all a single clone.
Really.
And somebody should look at it. And now you could look at the bacteria inside of it as well.
So, I mean, as far as we've read, I've also noticed that the same, that Oxalis pescum was a very sort of crazy invader in Europe. And from the study, I mean, there hasn't been a significant sort of statistical higher rate, but I've just noticed more diversity of endophytes among the Pescoprea plants. So it seems like it's even capable of absorbing even more. So, and there's been another recent study which detected a different bacteria, and it's called Azospirillum, which is also a nitrogen fixing endophyte, from these plants in Turkey. So it seems like they're capable of taking up or implementing whatever's available to them, which maybe adds to their sort of success as an invasive species. And the fact that these things are capable, the endophytes are vertically transmitted, so these seeds are released with all the potential that they could bring on.
You say that there are very few roots on the species you're working with?
Yes.
And the bacteria provide organic nutrients, but how did they get their water in the soil?
That's a good question. So within the Cape, the winter is known to be the wet season. So during the winter months, these plants, when it gets cold and when it starts raining, the leaves reduce and they make their seeds. So they just by the natural history, they're adapted to grow during the wet season. So I don't think they don't invest much in making roots, because I think water, in a limited sense, it's still abundant and easily accessible to them during that time. But they're very specially adapted to grow and do everything they need to do during those about 3 or 4 months, which is the peak of their flowering season, which is pretty remarkable if you think about it, because they have to, for the seedlings, they have to sort of germinate, grow, photosynthesize, and make a bulb, which is pretty spectacular.
They have asexual propagules as well.
Yes.
And is the bacteria transmitted through those propagules also?
We haven't looked at that specifically, but we have some very nice anatomical sections through those tiny bulbs, and they have the cavities as well. So if we're on the right track with this, it would be very likely that those cavities would then contain the bacteria as well.
Do you think there's no fungi?
That's a good question. We have looked at fungi, and we have, so far with our culture-based methods, we have cultured fungi, but the majority of those seem to be parasites or saprophytes, so they didn't have any direct beneficial association. And the common thing is to find mycorrhizal fungi, which inhabit the root material, but unfortunately they don't grow on agar medium. So we've sort of, in a way, missed that. But I have done a few staining techniques where you can use a specific stain that stains that fungi, and we've seen them, but we haven't sequenced them to identify them specifically. But also, these plants have very few roots, so it wouldn't be likely that they would rely on a fungi if they have so little root material.
Okay, thank you.